Hi, everyone. We're going to go over some categories of complex lipids and how they're made. I understand that this is a complex material. There is no way we can cover all these groups in a detailed and thorough way. As the point we had made in the previous videos is that lipids are very complex, so to go through all their biosynthesis is just beyond the scope of this class. But let's see some really basic ones. And I'll draw attention to specific points. So we saw how fatty acids are made. Okay, we saw obviously glycerolipids it's fairly straightforward. You esterified fatty acids into a glycerol backbone. Let's look at the glycerophospholipids. So again, we have two kinds of phospholipids with this phosphate group. Glycerophospholipids and sphingolipids. They both have this phosphate group. But here, what we're having with these phospholipids is the glycerol backbone. And the X is what is different. Okay, we have the two fatty acids, but the X here on this end, on the phosphate, it could be several things, but the most interesting ones for us at this point is the basics like ethanolamine, choline, and serine. They are related compounds. In fact, you can make one from the other in normal biosynthesis, and from serine. And we'll see how these are put together to make these phospholipids. So if you have nothing there and you just have a phosphate on the glycerol backbone, so one of the positions is phosphorylated, the other two positions on glycerol, the hydroxyl groups, are esterified with two fatty acids. This is phosphatidic acid. Okay, and you can easily make phosphatidic acid from glycerol. Okay, you have a kinase, you phosphorylate it. You have a glycerol 3-phosphate. We've seen this before, and then you simply esterify what's left, and then you get phosphatidic acid, PA, for sure. And you can go in a variety of ways to that outcome. You can, add and then reduce or reduce the hydroxyacetone phosphate first. So you start with dihydroxyacetone phosphate, not just with glycerol, but you can start straight up with a glycolytic intermediate, and you can reduce the two keto position, and then you essentially have phosphorylated glycerol, and you can acylate it and esterify these groups. So that's easy. That's the overall scheme that you can see here. The point of this slide is that if you have this configuration where you have only one acyl group on glycerol 3-phosphate, this is called lysophosphatidic acid, and that's from the breakdown of sometimes you get this if you actually start removing if you break down triglycerides or if you break down phospholipids, sometimes you get these lysoforms. Okay. Now, if you don't have the phosphate and you just have diacyl glycerol, okay, you can still join this group here, very straightforward, and that happens too. Now, let's look specifically at the major membrane lipids. And we have phosphatidyl serine, shown here, ethanolamine, and choline. By the way, choline, phosphatidyl choline, which is very abundant, about half of the phosphoglycerides are actually, or the phospholipids in general, which includes all these groups, is phosphatidyl choline. Choline is heavily methylated. It's got 3 methyl groups on ethanolamine here, on this amino group of ethanolamine. It's the same thing, except that it's a methylated version. And it turns out that phosphatidyl choline is the major sink of methyl groups in a cell, in a eukaryotic cell, right? Because there is a ton of them. I mean, let's put it this way. And these are the fatty acids that you see sticking out. Now, phosphatidyl ethanolamine, let's see how it's made. And the point that I want to make here is when you add these groups, let's say phosphoethanolamine, first you start with ethanolamine, then you phosphorylate, you have phosphoethanolamine, then, okay, you use again the nucleodidylation to activate these groups for biosynthesis. So we have seen already the same principle, activation. You take a trinucleotide, So you take an NTP, a nucleotide triphosphate, sorry, an NTP, and if it's at ATP, you can activate glucose like that, and you can make it starch if you're a plant. If it's UTP, you can uridilylate, as we saw, glucose and put it into glycogen in our cells. For these groups that you want to add to phospholipids, it's CTP that is used. Now, don't ask me why it's CTP in this case and not the other ones, and vice versa. I don't know, but the theme is the same. No matter what the nucleotide is, I mean, the theme of nucleodilation of something you want to activate to use in subsequent steps in biosynthesis is the same. Right? It's just the identity of the nucleotide that's used. And in this case, for these lipid prosthetic groups, it's CTP that's used to CD-delete the prosthetic group. In this case, ethanolamine. And then you use that with CDP ethanolamine now with diacylglycerol, and you get phosphatidyl ethanolamine, and then you're done. Which happens to be about 25% of all phospholipids. So it's a major one too. PC phosphatidylcholine is even more abundant. Now, a different version of these phospholipids is what you see here, where you don't actually have an ester, but you have an ether, okay, and then a double bond right next to it. These are called plasmalogens on one of the acyl groups. Okay, it's not esterified, but it's an ether bond here. Those are special. They're called plasmalogens, and the X here could be ethanolamine or choline. And why are they special? They are actually very abundant in particular brain cells, in specific brain cells, and it's just they're extremly abundant. Okay? So they have special interest just for the sheer abundance in brain. Phosphatidylserine is so why do cells go through this trouble? I mean, why do you have specific phospholipids? Why do we have all three? Let's say all of them. Well, it turns out that you can use them to tell which side of the membrane you are on, right? So on the cytoplasmic side of plasma membranes, we have a lot of phosphatidylserine. You don't find on the outside; you find on the inside. So you have the double bilayer, the bilayer, and on one leaflet, the inside one, the one that faces inside the cell, you have phosphatidylserine, but not on the other leaflet that faces on the outside. Right? Phosphatidylcholine, as I mentioned, is a very abundant and it's methylated. It's a major methyl sink. Choline is a major methyl sink in cells. How do you make it? Again, you phosphorylate like we saw with phosphatidylethanolamine. You start there with ethanolamine, phosphatidylethanolamine, and then you bring the CTP again and you nucleotidylate choline so it can be added to diacylglycerol and make phosphatidylcholine. Right? So it's the same idea again, the same principle. Likewise, if you have a CDP diacylglycerol, and then if you combine two of them together, you get something super special called cardiolipin. So I'm just going to skip the steps here and show you how it looks like, and you'll understand what I mean. So this is the phosphatidyl. This is one phospholipid, and this is the other, and it's joined by a glycerol, actually. Okay, and so this is called cardiolipin. It's very specific in mitochondria. Okay, it's actually a marker, the best marker available for mitochondria. If you know, how do you know you've purified something that's exclusively mitochondrial? You use cardiolipin. Is cardiolipin there and only there? Then you know you got a good mitochondrial purification. Why is it called cardio? Because of course there is a lot of mitochondria in the heart. When it was first isolated, people were just going to slaughterhouses, getting cow hearts or pig hearts, and it's rich in mitochondria, and that's where that lipid was found. Right? And now we get something really special: phosphatidylinositols. So these are not structural components of membranes. Okay, they're much rarer than the others. And first of all, how do they look like? You have CDP glycerol. Okay, that's a glycerol backbone here, and you have CDP. So you have added diacylglycerol, and that reacted, well, let's say, with CTP. You got phosphatidic acid actually, and then you CDP-delayed, so it's activated already. So you have CDP glycerol, and then you can use it in a reaction with myoinositol, which looks like this. Okay? And what you get is phosphatidylinositol. You release a CMP now, and you get, so you maintain this part, okay? And that's here. And instead of the CMP that you had, now becomes inositol, so that would be phosphatidylinositol overall. So these kinds of lipids, it is a phospholipid. There is it's a glycerophospholipid actually also. So there is a glycerol backbone. It's phosphorylated, and the X here is inositol. Right? What that does, it's not a structural role, but it's a signaling role. Each of these hydroxyl groups, it turns out, it can be phosphorylated by a specific kinase, and it can also be dephosphorylated by a specific phosphatase. So in a sense, you have a microprocessor. Each one of these positions, it can be phosphorylated or not, so binary, 0, 1, so you can play if you're computer savvy. 0, 1 here, 0, 1, 0, 1, 0, 1, 0, 1, you have all these combinations. And it is exactly how it is used in cells. And you can have this orgy of phosphorylations and dephosphorylations by specific phosphatases and kinases that go and pick up and put phosphates at specific positions and remove them. So the status, how many phosphorylations and at what positions you have them on this inositol of the phosphatidylinositol, you can relate that. Then you have many different outputs, 32 to be exact, okay, and that the cell can use to tell other components in the cell what to do. How important is this? Extremely important. Kinases and phosphatases are actually oncogenes and tumor suppressors. So these processes are involved in some fundamental cellular process. You're going to see some of these mechanisms involved in fundamental cellular process. You'll see some of them in the next slide with profound physiological consequences. So again, what you need to remember is that phosphatidylinositols are not really structural phospholipids. They are signaling phospholipids because the inositol component here, depending on its phosphorylation status, you can build a range of outputs, okay, based on this modification. So it does work like a microprocessor in the cell. What does it regulate? Well, the better question is, what doesn't it regulate? And you can have regulation of the cytoskeleton, vesicle formation and trafficking, various kinases, ion channels, you name it. Okay. All right, then we go to the last class of phospholipids, which are the sphingolipids, because they don't have glycerol, they have sphingosine. There is sphingosine, and then you have the fatty acid, and so on and so forth. You can stick various things onto them, and you get a variety of them. All right, so we're going to close with one special one that's kind of a side story here, but it's kind of important. If you break down phospholipids, and they can be broken down by molecules in let's say in the circulation, or by light if you have too much exposure, let's say to the sun, phospholipids will be broken down, and then eventually they're going to make it into arachidonic acid, and then they get oxidized by COX-1 and COX-2 to different enzymes and give you prostaglandins. Now it is these prostaglandins, okay, that regulate a whole variety of cellular responses, including inflammation. Right? So if you block formation of prostaglandins from phospholipid breakdown, okay, so this is not synthesis of phospholipids, this is actually breakdown of phospholipids. Okay. When they're broken down, it gives rise to this molecules, prostaglandins, which then regulate a whole variety of downstream events, such as inflammation. And this is arachidonic acid. This is a phospholipid breakdown product, which gets processed by this enzyme, cyclooxygenases COX-1 and COX-2, which happen to be inhibited by aspirin and other non-steroidal anti-inflammatories, such as ibuprofen, Advil, and so on and so forth. This is one of the oldest drugs used, and for for decades, we did not know its mechanism of action. So aspirin, that is, and other non-steroidal anti-inflammatories that I'm sure you've all used them, and you will be using them. You can buy them at any grocery store and pharmacy. And this is how they work. They block the further processing of phospholipid breakdown products to prostaglandins, which then in turn regulate inflammation. So you block cyclooxygenases, you block by these drugs, you will block prostaglandin formation, and you will block inflammation, which is what we care in this case. All right, so we'll stop at this point, and then in the last video, the next one, we're going to talk about cholesterol.